Does this work? Wow. It is complex, yeah. You know, there's a theorem about wires and speakers that does not redound to the benefit of the speaker. I'm very pleased to be here this evening. This is a rather awesome auditorium to be speaking in. I gather that there was some pretty heavy rock stuff here in previous years. I cannot imagine that a talk about the origin of life and its evolution carries the moment that a good hard rock concert does. On the other hand, I want to begin by introducing you to Manna. She's from Panama, carved about 50 years ago, but could have been carved 80,000 years ago. I've become deeply fond of her. Maybe some of you can come see her afterwards. What's wonderful about Manna, I know her name is Manna because I named her Manna, so it's sort of a cheat. Anyway, what's wonderful about Manna, I was up in Vancouver last week or 2 weeks ago, and this has got a ring in it. I wonder if somebody can adjust the sound down a little bit. Anyway, I was coming down the escalator at Vancouver Airport, kind of like this, feeling centered, which is what you're supposed to do when you have a figure that you can invoke your ancestors with. And there's these enormous totems coming up if you've been in Vancouver Airport. And both of us sort of looked at these totems. Something's going on here. And what's going on, after all, is something that roots us in our own past. A past that's at least 80,000 years old. And part of the interest of it is that we used to tell stories. We used to start once upon a time. Once upon a time. And I'm going to wind up wending my way back to what stories are about, being driven there in the oddest conceivable way from efforts to understand the deepest sources of origins of order and evolution of life on the planet. Almost everything that I'm going to... Mona, you can lie down now. Yeah. Thank you. Okay. She didn't like that. She thought... almost everything that I will tell you has to be seen by you as theory and speculation. I'm going to ask you to struggle with me about issues that are hard to understand. So let me tell you what I'm going to tell you. I will begin by telling you something about the standard view of the origin of life, and then I will raise questions about it. The experiments to try to get it to go just haven't worked. I will briefly tell you an alternative view of the origin of life. If I were to go into that at great detail, it would take an hour. So I'm not going to. I'm going to sketch its line. Because what I want to get onto is something that is completely mysterious to me, and I can begin it as follows. If you think of a bacterium like E. coli, swimming upstream in a sugar gradient, I think we're all willing to say of that bacterium that it's going to get dinner, or if it's Sunday, it's going, of course, to get brunch. But in any case, it's in some sense acting on its own behalf in an environment. I'm not yet attributing consciousness. I'm just saying that we're willing to say of the bacterium that it's hoofing it for dinner. So I want to call an autonomous agent a system that can act on its own behalf. We all got dinner either before or you will after you've come here. But I think we also want to say that a bacterium is just a physical system. What does a physical system have to be so that it can act on its own behalf in an environment? What does a system have to be able to be an autonomous agent in an environment? And the attempt to answer that has led me down the strangest of pathways, which is going to lead back to stories at the far end of trying to be science. So pay attention, do not believe, but come along with the struggle. If any of you want to find any of this material, I wrote what is now a preprint at the Santa Fe Institute called Investigations. It started as follows. About 1994, I sat down with full brazen whatever, and you know Wittgenstein, the philosopher, wrote Philosophical Investigations, and many of us looked at Wittgenstein at some point in our life, and I began this thing and I said, Investigations, Stuart Kauffman, 1994. Then I chickened out and I said, confidential, lots of BS below. So 400 pages later, I had shortened the thing in a subsequent version to something that's 200 pages, and it's now in press or in preparation at Oxford. Okay, enough general introduction, enough forewarning, but be forewarned, we're on to strange turf almost immediately, but not quite immediately. I want to talk to you about the standard view of the origin of life, and in fact I should set the setting. 200 years ago there was not a problem about the origin of life. Life obviously came about spontaneously. Look under a rotting log and it was coming forth all the time. Kind of a gooey, messy stuff, but maggots would come crawling out of nowhere, and so it was obviously life just started. Pasteur really is the villain because he demonstrated in the last century that life only comes from life. In his brilliant experiments where he took a broth that was sterilized and made an S-shaped neck with fluid in the S-shaped neck that prevented air from getting from the outside to the inside, and showed that the broth remained sterile, and concluded life only comes from life. So once that's true, we've got a problem. If life only comes from life, there seems to be a problem getting life started, of course. Well, the next major step that was taken in the origin of life's struggle was in 1954 when Watson and Crick published their famous paper on the structure of DNA, and you all know the famous double helix. One strand is called Watson and the other strand is called Crick. It was very good that there were two discoverers. I mean, what would we do otherwise? It couldn't have been Frank Crick. Wouldn't have worked. Anyway, as you know, there's this double helix, and you all know there's 4 nucleotides. In DNA, it's A, C, T, and G. And in RNA, it's A, U, C, and G. And I think you all know that C's bind G's, and A's bind T's, or A's bind U's, which means that if you come down the Watson side of the double helix, and you were to see GGG, CCC, AAA, UU, well, TTT for DNA, you would know immediately what the corresponding nucleotide was on the Crick side. For the C's would make G's and the so on. So you have the idea, you understand it. And in fact, it works, and in fact, the proper double-stranded DNA molecule as a double helix has this exquisite twofold rotational symmetry, so you can drive an imaginary line down its center, and it's rotationally symmetric about it. And anybody who's looked at that molecule has concluded, my golly, what a gorgeous structure to have life start with. Because one strand can carry an arbitrary sequence of nucleotides, hence genetic information, and it'll specify the other side. Watson will specify Crick, and Crick will specify Watson. Same true for RNA. Now, if only we could get one of these thingies to replicate in the absence of any enzymes. The enzymes are going to be important to our story. Remember that chemical reactions are reversible. A goes to B, B goes back to A, and at equilibrium, the bulk rate of change of A to B is equal to the bulk rate of change from B to A. You all remember when you were kids in high school and you took sodium chloride and put it in water, and if you had enough sodium chloride in water, you got some sludge at the bottom and an equilibrium solution of ions up above it. So that's an example of that equilibrium. Enzymes speed up the approach to equilibrium, but they do not change the equilibrium of a reaction. Anyway, so the idea is we have a single-stranded RNA. RNA molecules are oriented polymers, 3', 5'. And the idea that people have been in love with for 30 years, I am too, is that you have A's and C's and T's and G's, and each one lines up the appropriate, well, AU, C and G, and each one, picked by the different colors, lines up the appropriate other nucleotide by Watson-Crick pairing, standing free from one another, and then somehow these get zipped together to make the appropriate 3', 5' bonds, so that Watson makes Crick, then Crick and Watson fall apart, and Crick now lines up things to make a second Watson, and Watson lines up things to make a second Crick, and so you have a reproducing molecular system. It's an absolutely beautiful idea. There's nothing wrong with the idea. It's exquisite. The only problem is it doesn't work. It doesn't work for good chemical reasons so far. The fact that it's not worked so far does not mean it will fail to work sometime in the future, but what happens is the following. The RNA molecule needs to have 3', 5' bonds between the adjacent bases, But in fact, thermodynamically, it's much easier to make 2' 5' bonds. And that happens to make a ladder side that kinks up badly, and it's hard for it to have the twofold symmetry, rotational symmetry. So that's problem one. Problem two is you just can't get this to work with all 4 nucleotides. What does work is to make the bottom strand with only C and G, but a lot more C than G. But then necessarily, if Watson is a lot more C than G, the new Crick will be a lot more G than C, the other way around. And RNA strands that are rich in G, in guanidine, tend to wrap up and stack the G's on top of one another, and they just precipitate to the bottom of the test tube and kind of wiggle their nose at you and say, nah, 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 I won't do anything. And that's what happens. It's sort of tough. Okay, so it hasn't worked. Now there's a new standard view, and it's called the RNA World view. And the idea is that at some stage early in the evolution of life, all of the business going on inside of a self-reproducing molecular system was based on RNA. And it comes with the discovery that there are RNA molecules that can act as enzymes. They're called ribozymes. They were discovered in group-run introns in tetrahymena, where a long RNA molecule snips out a little internal sequence called an intron, and the RNA molecule in question can actually act as a little catalyst and do the outsnipping. Of course, the German for that is this ausgeschnippt. Okay, it comes ausgeschnitten. I don't know what it is in German. Anyway. Okay, so das ist ausgeschnippt und that's an enzyme. So now the idea is to try to evolve an RNA molecule that will be able to be what's called a polymerase. And the idea of a polymerase is it's an enzyme that can ride along this sequence, the Watson sequence, with an arbitrary sequence of nucleotides and take the lined up free nucleotides and appropriately snip them together to make the Crick strand. To do that, since there are 4 nucleotides, there are 16 possible reactions, and the enzyme has to distinguish the right one of the 16 at each moment. So it's pretty hard. But meanwhile, Jack Szostak at Harvard Medical School is busy trying to evolve such an enzyme, and I think Jack's got a reasonable chance of succeeding. Is there too sibilant a s in this? Or does it sound okay? It's okay. It's an echo coming back at me. Okay. So Jack may succeed, and I want in the transition right here to tell you something that's going on that will change your lives in the next decades. It has to do with a whole new area called molecular diversity or combinatorial chemistry. The standard way 10 years ago of cloning a gene of use was to clone the insulin gene from a human being, the gene that makes the protein, snip it out and put it into a bacterium. The bacterium obliges by making insulin, and so we've got insulin, and whoever was the company that did it gets rich. In fact, gets filthy rich. It is lucre beyond galore, and it's just fine. Meanwhile, there's a whole new idea, and the new idea is roughly the following. Life in the past 4 billion years, or 4.5 billion years, can have only tried a tiny fraction of the possible proteins. We'll get back to that later. What about all the proteins that have never been tried by life any time in life's history? Maybe there's nifty proteins out there able to do things. So the new area is called molecular diversity, and what one does is makes literally trillions of more or less random DNA, RNA, or proteins, and tries to fish out molecules capable of doing anything you want. So an example would be the following. Women have estrogen receptors. The estrogen receptors have been cloned. Obviously, estrogen binds to the estrogen receptor. Think of estrogen as a key and the estrogen receptor as a lock. Now take 100 million random small proteins and try them one by one and see if any of the novel proteins happen to bind to the estrogen receptor. If so, you have a second key that fits the same lock that estrogen does. So it's plausible that estrogen and this new molecule are similar to one another, since they both bind the estrogen recepter. A fortiori, you're on your way to a potential drug that may mimic or modulate the effect of estrogen. This is now working and working brilliantly. People have now cloned on the order of 10 to the 14th, which is 100 trillion random proteins on the surfaces of viruses. It's called phage display. People are busy fishing out things that bind estrogen receptors and a lot of other receptors. So we're on our way to drugs. Had I started with the hepatitis virus and taken an antibody molecule that bound to the protective molecular bump on the hepatitis virus, and then I found a peptide that bound to the antibody molecule, I can think of the bump on the hepatitis virus as a key, the antibody molecule as a lock, and the second peptide that binds to the antibody molecule is a second key binding the same lock. I'm on my way to a vaccine that looks like, because it looks like the hepatitis virus bump. And notice that I can try to do that without actually having the hepatitis virus. I can try to do it with just the antibody molecule. So I can try to make a vaccine against an unknown pathogen. Or, since the way enzymes work are to bind a transition state of a reaction, see, there's a substrate and a product state of a reaction in this sort of measurably distorted form called the transition state. The way enzymes work is they bind to the transition state and stabilize it, and that speeds up the reaction. That suggests that it's going to be possible to evolve new enzymes. Among the bizarre implications of this is that there's, just like there's a number of skeleton keys, and the number of skeleton keys is less than the number of keys, there's good grounds to think that there's about 100 million to a trillion effectively different molecular shapes. And if that's correct, we're going to be able to create universal toolboxes of molecules to do pretty much any reaction we want. So that's not just on the horizon, that's happening now in drug companies and biotech companies all over the world. What's going to happen in the next 2 decades, and part of what I want to talk about tonight, is the creation of systems of novel molecules. And I think that within decades we are going to create self-reproducing systems of molecules that will be autonomous agents, what I want to talk about tonight. And we stand therefore on a threshold of a scientific revolution that will equal easily the computer revolution. The biosphere is built out of 10 to the 14th different kinds of molecules. We can manipulate that many now in a test tube. Okay, let me show you what does work. We started at about 1:45 after Terry's interminable introduction. But, by the way, I've gotten to know Terry Bristol pretty well in the last 4 days. He is a really neat, really smart guy. And it seems to me that he's putting together lecture series that are really fun for you all. And I'm enjoying this. Didn't tell you I was going to do that, Terry, did I? Okay, here's what has worked. People have actually made reproducing molecular systems. And here it is. I've got a 6-nucleotide sequence in the DNA. Von Gederowski has done this in Germany. So this is a single-stranded DNA, GGG-CCC, a hexamer. And it lines up 2 trimers, GGG being lined up by CCC, and CCC being lined up by GGG. So the hexamer holds the 2 trimers adjacent to one another and therefore acts as a surface catalyst and speeds up the reaction that eliminates the water there and glues these 2 sequences together. And when that reaction has happened, since this is an oriented polymer, 3', 5' GGG CCC, the top strand is going in the other way, 3', 5', 3, 5, and when you do that you get GGG CCC, which is identical to the bottom one. It is a self-reproducing molecular reaction. It is autocatalytic. It works. Now the difference between this and this is the top one is supposed to have an arbitrary sequence of nucleotides and carry arbitrary genetic information. This thing is a ligation reaction with a specific hexamer binding 2 specific trimers that are complements by Watson-Crick pairing and gluing them together. But we have the world's first self-reproducing molecular system made by us rather than by 4 billion years of evolution. The really staggering result was accomplished last year and published by Reza Ghadiri and his colleagues. It's Lee et al. In Nature, August 17th. Reza made the world's self-reproducing protein. See, a protein doesn't have this beautiful 2-fold symmetry of the double helix. A protein is a linear string of amino acids, 20 in kind, that then wads up into some shape like hemoglobin, and it's not obvious how it would specify a complementary structure, which is why people have thought that proteins can't have much to do with anything. So what Reza did is he took this 32 amino acid sequence protein. It folds into an alpha helix made famous by a townsman of yours named Linus Pauling. And then, as Linus also proved, a helix can fold back on itself to make a coiled coil. Reza and his friends reasoned, that's the word you always use, they reasoned. Actually, they didn't reason, they lucked into it. But after the fact, they reasoned, okay? And they say it as follows: we reasoned, okay, so they reasoned that, or lucked into the fact that the open helix, since it folds on itself, might have the property that if you took a second copy of the protein and cut it into two fragments, 15 and 17 amino acids long, that the 32 amino acid jobby might be able to line up the two fragments and hold them together just like it happens up here, and then glue them together by surface catalysis. And it works. So you take the 32 amino acid sequence and it can glue together the 15 and 17 amino acid subfragments. It's a ligation reaction and it's autocatalytic. So if you start with a few of the 32 amino acid proteins and there are an awful lot of the substrates, 15 and 17 amino acids, it cranks away and it's a self-reproducing molecular system. So Reza's done it. Now this is wonderful because Reza and Phil Anderson, who's a Nobel laureate, and Andy Ellington, who's worked with Jack Szostak, and I and others, have now gone to NASA for about $5 million to try to make self-reproducing molecular systems. And since Reza's already succeeded, we're bound to succeed. I mean, it's the best possible type of experiment. We'll just do it over and over and over and over again, and we'll say, see? In my second encounter, or my first encounter with Vice President Gore, it came to this really charming moment. It was sitting around a table with Steve Gould and Lynn Margulis, who you've met here, and we were discussing the Mars rock and what it meant. And in any case, Gore was extremely polite to me, and he said, Dr. Kauffman, don't you have this view that if you have a complex chemical soup, it will just create self-reproducing molecular systems? And I said, well, yes, sir, I do. And he said, well, doesn't that just seem sensible? And I said, well, I think it seems sensible. It would be very nice if there was some evidence other than computational evidence. And he said, yes, but wouldn't anybody who was sensible just think that was right? And I said, Mr. Vice President, I've waited for a long time for confirmation, and with your permission, sir, I'll use your support to bludgeon my enemies. I've actually never gotten to say this in public, but it was such a nice moment. Anyway, I'm going to vote for him, obviously. Anyway, I'm going to show you the first double whammy. Here's a protein A that catalyzes its own formation, and a protein, well, let's take Reza's system. Let's imagine that we have Reza's molecule that catalyzes its own system, like we've just seen. And imagine we had another protein B that did the same thing. I just want to use some of this molecular diversity magic to fiddle around with who catalyzes what reaction. So I want to make a system in which here's A and its two fragments, and here's B and their two fragments. But I want to have A catalyze the formation of B out of B's fragments, and B catalyze the formation of A out of A's fragments. These lines represent reactions, and the blue thingies represent catalysis. Gudrun von Katarowski has already done that with DNA. Reza and friends are not too far from doing this with proteins. Suppose we can do that. Here's what I want you to see. This is a collectively autocatalytic system. There's no molecule in this little thingy in which the molecule itself catalyzes its own formation. A catalyzes the formation of B, and B catalyzes the formation of A. It is collectively autocatalytic. There's no mystery here. Look at it. You can see it. Okay? I think everybody can see it. And if you count, I'm going to assert it. Okay? See? Now, if we can make two proteins where A catalyzes the formation of B from B's fragments and B catalyzes the formation of A from A's fragments, why can't we make three? A making B, B making C, and C making A? Or why can't we make 1,000 proteins, each catalyzing the formation of somebody else out of fragments in some complex, rich web of catalysis? Well, I think we can. Now, I'm not going to show you a 15-minute digression about buttons and threads. I'm just going to tell you, because I want to get onto the autonomous agent part of this story, I just want to tell you the following thing. If you take, well, I will show you buttons and threads. If you take 10,000 buttons on the floor, it's remarkably the same in a way. Yep. I know I'm stupid. So here we are with 20 buttons, and I'm connecting them at random by picking up pairs of buttons and tying them with red threads, even though they're black. And as you do that, and you connect more and more buttons just at random, every now and then hoist a button and see how many buttons you picked. Well, if you have thousands of buttons and only a few threads, most of the time you pick up a button, you won't pick up any button, but every now and then you'll pick up a pair of buttons. Then as you add more threads, you'll get little triplets, and then bigger structures and bigger structures and bigger structures and bigger structures. And the main intuition here is the following. That is, the ratio of threads to buttons goes up. You get little clusters, then bigger clusters, then bigger clusters, and then a few more threads tie together the middle-sized clusters into a vast cluster called the giant cluster. That's very technical. It's called the giant cluster, sort of mathematical. And so you have, if you look at the ratio of the largest cluster as a fraction of the ratio of threads to buttons, looks like that. At first they're small and then it jumps up. And it jumps up when the ratio of threads to buttons is half as many threads as buttons. And the deep reason is the number of ends of threads is twice the number of threads. And when the ratio of threads to buttons is 50%, the number of ends of threads is the same as the number of buttons. And you get this big giant step size thing. So some mathematics later, the same property holds in complex chemical reaction systems. And I just want to show you what it looks like at the end. If you make an increasingly diverse chemical soup, and don't lose your transparencies, at some diversity in your chemical soup, there's so many molecules and so very many more reactions that if the molecules can be substrates and products of reactions, but the molecules are also candidates to catalyze the reactions, like random proteins are for catalyzing reactions, then at some diversity you're bound to catalyze so many reactions that you get a big connected web of catalyzed reactions, at which point you almost certainly have a closed autocatalytic set in which a whole bunch of molecules make one another. And it doesn't matter that that one's backwards. You see? I showed you. Same thing. Okay, so that's all I want to say about the origin of life, except to say the following. I know that's comedic, but I'm so puzzled about what comes next that I want us to get into that. If this view is right, though, and either NASA gives us some money to do it and we succeed, or if the Mars rock turns out to have life on it and it's radically different than Earth life, if these views are right, so let me sort of stick with what I've told you, then it is not more than a decade or 10 to 20 years before we will be making complex soups of molecules that in fact are collectively reproducing sets of molecules. We will be making, at least in this sense, life forms. Now, I'm not prepared to call them life forms yet for a moment, and I'll show you because I have to tell you what an autonomous agent is. We are on the threshold of just a vast revolution. If we either make or find novel life forms unrelated to Earth life, in the sense no DNA, no RNA, no code, for example, we're going to have to reckon that the emergence of life may be much more probable than we have thought in the cosmos. And in fact, the point here, and that's why I called At Home in the Universe, At Home in the Universe, is that I think that life is based not on template replication, but on something very much deeper, based on catalysis, chance, and number. So that it's a natural expression of the universe to give rise to complex soups of molecules which self-organize into systems that are reproducing. I don't know that that's true. I strongly believe it. I think we will demonstrate it experimentally in the near future. That will not answer the question of how life started on Earth. Maybe it got here from Mars rocks or little green men. That's an historical question. But if it's the case that the emergence of life is plausible and easy chemically rather than hard chemically, then we are a natural expression of a non-equilibrium universe. We are not rare. We're to be expected. Other life forms are out there. In this galaxy, there's 10 to the 11th stars. Probably 10% of them to 1% of them have solar systems. And very crude calculations about the number in a habitable zone and the probabilities that life starts in such a case suggest that there may be something like 10,000 to 100,000 planets with life in the galaxy. The closest one being something like 30,000 light years away, sort of scattered at random across the galaxy. So who knows if that's right? But if it's right, not only are we not alone, but life is a natural expression of the cosmos. And we, therefore, are natural unfoldings of a non-equilibrium universe. And I want to continue with this theme as we go along, because we are at a spot that, in fact, our Vice President spoke about. He asked, what are the spiritual implications? Now, if we find such a thing. And I think that they're just vast, and I want to come back to that. Well, now I want to come to this molecular autonomous agent. We were on almost firm ground. Almost nothing is firm ground for the rest. I've been too slow. If I went over 5 to 10 minutes, would you kill me? Or stay? Would that be okay? Okay. You stay 30 minutes extra. Well, I won't do that to you. Okay. I want to give you a tentative definition of a molecular autonomous agent. I do not know that it's right. I think it's useful. It keeps revealing itself to me as more and more fecund. But what follows from that is that it's fecund, maybe. It doesn't follow that it's right. I want to describe what is a physical system able to act on its own behalf, the E. coli I talked about, bacterium. So let me try it on you. I'm going to talk about a molecular autonomous agent. By autonomous, I don't mean that it's not in an environment. I mean that it can act on its own behalf. So I'm going to say that a molecular autonomous agent is an autocatalytic system carrying out one or more thermodynamic work cycles. I had to learn what a thermodynamic work cycle was 2 years ago. I've been in the Department of Biochemistry and Biophysics at Penn for 15 years and had avoided learning what a thermodynamic work cycle was. I mean, there's all these competent people who know that. Why learn it? So I'm going to tell you. So I have to tell you a little bit of thermodynamics. What the heck? Wouldn't hurt. This drawing is really important because the smudges here are terrible. So ignore them, please. Now, I'm going to draw a chemical reaction here. Here's the 15-mer and the 17-mer and the 32 amino acid sequence. This is just Reza-Ghadiri's little system. And I'm drawing the reaction with the substrates and a set of lines representing the reaction. So this is a ligation reaction. Remember that reactions are reversible. They go in both directions. And I'm drawing the 32 amino acid sequence with this blue line, meaning that it catalyzes its own reaction that glues it together. So that's Reza-Ghadiri's reaction. The fact that reactions are reversible are essential because it defines chemical equilibrium. So if we start with excess substrate, 15 and 17 amino acid sequences, the reaction will flow in the direction of creating the 32 amino acid sequence. But we could also start in a direction in which we had a lot of the 32 amino acid sequence and no 15 and 17 amino acid sequences. So we could start displaced, as you learned in high school chemistry, to the right. So if we had all 32 amino acid sequences, there would be a reaction by which cleavage would happen, giving rise to 15 and 17 amino acid sequences. Equilibrium happens when the forward and the reverse rates are equal, as I think you all remember. So let me describe that equilibrium here, and it's when there's a stable ratio of the substrates to the product. The physicists and the chemists came up with this nifty definition of it called the free energy. It's called Gibbs free energy. You think it's named after Gibbs. It's not. It's named after free energy. Gibbs was an interloper. Okay. Now the idea is that when a chemical reaction is approaching equilibrium, it goes down in free energy, reaching a minimum at equilibrium, and the flow towards equilibrium is a spontaneous chemical reaction. It gives off free energy, so it's called exergonic. And the idea is that only those reactions that are exergonic happen spontaneously. But you can drive a reaction beyond equilibrium if you couple it to some other chemical reaction, which itself gives free energy, gives exergonic free energy, and then you drive the chemical reaction beyond equilibrium, so you drive it in a way that adds free energy, and it's called endergonic. You're pumping energy into the system. And in fact, if you drive it endergonically in this direction, you'll wind up with more 32 amino acid sequence than you would at equilibrium. So you've gotten an excess amount of the reproduction to happen. Okay. Now, is Reyes' system an autonomous agent on my hoped-for definition? And the answer is no, it's not. It is merely an exergonic reaction. There is no work cycle happening here. It's just exergonic. Now, you have to sort of struggle to catch up with this and follow this, but there's no equations, and there's abundant confusion. So, as the Irish say, if you're not confused, you don't understand the situation. So we'll proceed on that basis. Well, here's the famous Carnot cycle. By the way, there's an interesting reason why Carnot invented the Carnot cycle. Although I don't think he called it the Carnot cycle. He probably called it the cycle, and somebody else called it the Carnot cycle. The reason is he was really peeved at the Brits for the Battle of Waterloo. You remember that the French didn't do too well. And if you don't remember, Carnot will tell you. In any case, what he was angry about is that the British had pumps, and they used the pumps to pump water out of the mines. So they go down in the mines and they could mine coal and iron that they could use to make cannon, but they could also use the coal and iron to make pumps that they could then use to pump out the mines so that they could make coal and iron to make pumps. So it's an autocatalytic set, right? Anyway, Carnot was extraordinarily peeved at this, and he invented the Carnot cycle. So he was trying to understand, how do you get work out of heat? And he had this really bright idea. He had this abstract version of a machine. We have two temperature reservoirs. Temperature 1, which is hot. You can tell because I had red to point at it, so it's hot. And temperature 2, which is cold, and you can tell because I had blue to point at it, which is pretty good for a biologist. Then there's this thing called the cylinder and something called a working gas and a piston. Working gas is not a political statement. It's a gas. Now, there's something else that they don't tell you about in the physics textbook. There's a red handle. It's got to be red or the whole thing doesn't work. And here's what happens. You start with the piston pushed up in the cylinder sort of vigorously, so you've compressed the gas and it's hot. Then how it works is I, Stuart Kauffman, an amateur physicist, I pull on the handle and I pull the working gas over into contact with the hot reservoir. And then I allow, for example, I release any pressure on the piston and I allow the piston to expand. The gas expands so it gets cool. But because it's in contact with the hot reservoir, the cooling gas sucks heat in and therefore stays at the same temperature. And that is the isothermal part. This is pressure, temperature, volumes of the volume. This is the isothermal part of the power stroke. Then, and they don't tell you this, I, Stu Kauffman, amateur physicist, push on the handle and push this out of contact with the hot reservoir, and I let the piston coast outward with the gas expanding on the end of the power stroke, and since the gas is expanding, it cools, and that's the adiabatic expansion part. That's the end of the power stroke. Now, unbeknownst to the physicists, I come over and I, Stu, push on the handle and I push the working gas into contact with the cold reservoir. Then I push on this thing down here that pushes on the piston and compress the gas, but the heating gas can dump the heat into the cold reservoir, so it's easier to compress, and that's the isothermal part of the contraction cycle. Then I pull on the handle, pull it out of contact with the cold reservoir, keep pushing on the cylinder, recompress the gas, and heat it, and I'm back to the start. So that's the work cycle. Pretty neat. And I got it right. By the way, if you run it the other way, this thing is pumping heat from the cold reservoir to the hot reservoir, and it's a refrigerator. It's all sort of strange, but it was worked out in the last century. Okay. So now I'm going to make a hypothetical, theoretical biologist's conceivable paper chemistry autonomous agent. Once I've shown it to you, you'll see it, and my bet is that within 10 years we've made one. So here it is. Here is Gunther von Kederowski's little hexamer replicating system. Here's the 6-mer and the 2 trimers. Now the way biochemists draw chemical reactions is they draw a forward reaction, gluing the 2 trimers together to make a hexamer, and then they draw the reverse reaction by which the hexamer breaks back down into the trimers. So I think you can all see the way biochemists do it. As before, I'm going to let the hexamer catalyze the reaction that glues the 2 trimers together. But I want to couple this exergonic reaction in my magical chemistry to a free energy source that's going to drive the reaction endergonically, this reaction, to make excess hexamer. And what I'm going to do is I'm going to use PP, which is pyrophosphate. Pyrophosphate breaks down to 2 monophosphates with a big drop in free energy. It really loves to break down. And the way I'm going to do it is I'm going to couple it so that when this enzyme, the hexamer, catalyzes the reaction, it does so by coupling the breakdown of pyrophosphate to monophosphate to the synthesis of the hexamer. This happens all the time in your cells. Not this reaction, but this kind of coupling. So what happens is the following. When the enzyme is turned on, there is a flush through, and the excess free energy drop going from pyrophosphate to the 2 phosphates winds up with excess synthesis of the hexamer. So we've got excess replication of the hexamer. Can everybody see that? I think everybody can see that. We've just made extra. We've just driven it endergonically beyond equilibrium. By the way, you know that handle that I was pushing and pulling? How's that actually done in a real machine? Well, Stu doesn't stand there doing that. Somebody comes along and says, you know, we could replace you, you stupid clod, with gears and escapements and widgets and so on that couple everything together. And there are gears and escapements and widgets and flywheels that do that. And I'm going to come back to that because I'm going to have some gears and escapements in here in a second. That's essential to the story. Anyway, having flooded through and made the excess synthesis, now what I want to do is I want to turn off this reaction and I want to turn on the reaction that's going to go back because I've broken down pyrophosphate to monophosphate and I want to restore the pyrophosphate. Now this is displaced from equilibrium. I have to eat from the outside the 2 trimers, and I'm going to invoke a new hypothetical reaction called a redox reaction carrying something F to the ionized forms F plus and E minus with an even bigger free energy drop that way. So I'm eating F. This is an open thermodynamic system. I'm not cheating. It's not perpetual motion. So I need an enzyme, and I'm going to just let CCC be that enzyme, and I want to turn it on. And when I turn it on, then there's the back synthesis that restores pyrophosphate to its former level with the breakdown of F to F plus E. The cycle of PP to pyrophosphate to the 2 phosphates and back is like the cycle of the working gas that we just saw. The working gas expanded, then contracted. The concentration of pyrophosphate being up, going down, and coming back up is identical spiritually to the cycle of the working gas. Meanwhile, the handle pushin' and pullin', or the cogs and gears, I'm going to do by invoking a little more hypothetical chemistry. I'm going to imagine that phosphate feeds back and turns on the enzyme, this enzyme, so that when phosphate's low, the enzyme's inactive, but when phosphate's high, the enzyme's active. That's perfectly standard biochemistry. That happens in the glycolytic cycle in your cells, where the product of an enzyme called phosphofructokinase feeds back and turns it on. And what happens is, the product is formed slowly, then feeds back and activates the enzyme, turns on the enzyme, and then there's a flush through from substrate to product. This is known very elegantly in the literature as a toilet flush model. So you get the idea. So here's what happens here. This happens, there's not much phosphate, the enzyme is inactive, there's a slow leak through, then phosphate builds up, feeds back, opens up the enzyme and activates it, and there's a flush through. As soon as that happens, the phosphate's around, but now pyrophosphate's gone. I've invoked pyrophosphate as an inhibitor of this reaction. I should have a little minus sign. When the pyrophosphate is gone, the inhibition is gone, so the back reaction happens. So those are my gears and cogs. Meanwhile, because I've turned off in the back reaction when I remake the pyrophosphate, I've shut off this enzyme. Therefore, the enzyme is not busy breaking down the newly synthesized hexamer back up here. So I get a flush through excess synthesis of the hexamer, then I turn off the enzyme that would have broken it back down, namely the hexamer, because pyrophosphate is restored and there's no phosphate. And so the back leaking here is very slow, and I wind up with excess pyrophosphate. And meanwhile, I've restored my pyrophosphate levels. So that has gone through a work cycle. So I've shown you one. So we can make them. In the next 15 to 20 years, we will surely make an autocatalytic system that can do at least one thermodynamic work cycle. If this is silly chemistry, it doesn't mean we can't do it. There's a lot of things that are going to follow from this. The first is that by having excess hexamer around, I have stored free energy in my system, and I can use it later to error correct. Okay, now we're going to get into a bunch of puzzles. I guess with your permission, I'll make it to 9 and then pump. Okay, I want now to get to the puzzles. We've already seen in an autocatalytic set, and I showed you one, the first one where A made B and B made A, the notion of closure in a space of catalytic tasks. It is an objective property of a set of molecules that A catalyzes B and B catalyzes A, that it's collectively autocatalytic. There is a holism, but there's nothing mysterious about it. It's true. Okay, now I'm going to move us towards the notion of propagating work and closure in a space of work tasks. These are new notions. I may be absolutely nuts, so be careful. So here is the naive biologist's view of physics. Here is a cannon. The evidence that it's a cannon is that it says cannon. This is a cannonball. The evidence that it's a cannonball is given by cannonball. And the cannonball is fired from the cannon, and it flaps into the dirt, and it makes a hole. There's it is, this hole. And it makes hot dirt. Okay. Sounds like a waste, but in any case, that's what happens. Now, I could have avoided the hole, which is work done on the soil, by making this thing a really big steel plate. And the cannonball thwacks into it and bounces along the surface and gradually rolls to a stop. And I have a vibrating plate of steel, so it's a hot steel plate, but there's no hole. So I've transformed the kinetic energy of this cannonball into the thermal vibration motions of the plate. Okay, now I want to show you an extremely sophisticated device, which I built. There's the cannon. Here's a cannonball. This is a paddle wheel, as you can tell by the label. It's in fact a sturdy paddle wheel. The only way you know that it's a sturdy paddle wheel is that I'm telling you. You can't quite tell. Now, attached to the paddle wheel is a rope that goes down, and you can tell it's a rope, into the water, and it's attached to a bucket, and the bucket is filled with water. And there's a pipe here. Here's the way my widget works. When the paddle wheel is hit by the cannonball, the paddle wheel spins. You can see I've done something wrong. I really wanted it to spin the other way, but the pipe was in the wrong way. Anyway, it spins, and the rope winds up on the axle. It should be going the other way, as you're going to see. But in any case, the bucket comes up, and it kind of gets trapped underneath, which is not what I wanted. And it... But I'm a biologist, you know, what do I know? Anyway, the water is tripped into this pipe. You can tell it's a pipe for the obvious reason. And the water flows down the pipe, and it opens a flap valve, and it comes out and it waters the cornfield. Corn grows. I think it's really terrific. I'm very proud of it. Well, what's the difference between this thingy and that thingy? There's the same release of kinetic energy. It's just a cannonball. I want to call this propagating work. Now, there's physicists in the audience. Bear with me because I am puzzled about this. But you see, it has to do with the fact that work is a curious notion. I know that work is a curious notion because Phil Anderson's a Nobel laureate in physics, and we were out for an Indian dinner 2 years ago, and I said, Phil, work is a curious notion. And he said, yes. What's that? I rest my case. Okay, so what's the difference between the top one and the bottom one? The bottom one, there's a lot of macroscopic changes happening in the world. There's paddle wheels whirling and ropes and so on and so forth. So I want to try to talk about the notion of propagating work, propagating macroscopic changes in the world, and it's happening here. By the way, this already shows you something interesting. This is quite a Rube Goldberg, if you all remember what Rube Goldbergs are. Why does the Carnot cycle work in a cycle? Well, the answer is it's real easy to make a widget that comes back to a state that it's been in before cyclically and can take some more energy as an input and then produce some work going around again. This is a hard thing to find out in the world. I mean, they don't crop up that often. In fact, that may be the only one. Okay, which brings us to what is work? Now, the people doing quantum cosmology and quantum physics will tell you that the notion of work is passé. And I think they're referring to their own work habits. I won't defend that. Anyway, work is a funny notion, and I want to get you to feel why it's funny. So one asks one's physicist friends, tell me, what is work? This is a strange way of approaching a physicist friend, and you're likely to get slugged in the nose. But if they answer anything, they'll say, fool, work is force acting through a distance. What they've got in mind is you're standing out on some ice, and you've got a hockey puck and a hockey stick, and you go thwap, and the hockey puck goes that way. So you've done work on the hockey puck, which is true. So work is force acting through a distance, but when they say that, they actually somewhere have picked a direction. It's the puck going that way. Who picked the direction? Where did that come from? I just wanted to make you feel a little puzzled. Maybe there's an answer to this. But if you blow up a balloon, you get the balloon blowing up, and so the directions are all directions. But somehow, who picked that? Well, maybe there's a way around this because something changed, but just hold that. There is the notion of direction that got snuck in to the definition of work somewhere, somehow. Another way of playing at this puzzle is the following. An isolated thermodynamic system, closed isolated thermodynamic system, can do no work by the first law, conservation of energy. But if you partition it, one part can do work on the other part. For example, maybe it's a gas and you put in a membrane and there's a little more pressure on one side than the other and the membrane bulges. So the right side has done some work on the left side. Well, that's dandy. But who picked the partitioning of the system? Where did that come from? Now, the third way to point at this puzzle, and I'm not sure exactly what this puzzle is to use Atkins' definition of work. This is in his book on the second law, and he says, work is the constrained release of energy. So he says, work is a thing itself. Well, what does he mean? He says, well, let's take the cylinder and the piston. Here's the cylinder, here's the piston, there's some grease I haven't shown you, there's some hot gas, and the hot gas expanding does work on the piston. Well, it does. I mean, that's what makes the choo-choo train go round and round. So the constraints are the cylinder and the piston, and somebody had to squirt the gas in there. That is to say, work was done to build the constraints, the cylinder, the piston, and the assembly. Just like my Rube Goldberg in the past thing, I'm the one who made the sturdy paddle wheel and tied on the rope and went hustling around. So it took work to make the constraints. One is led to at least say to oneself the following puzzling but maybe accurate phrases. I don't know what to make of this. Something like, no work, no constraints. No constraints, no work. Stay puzzled. What does a cell do? A cell does it. It's going to get more and more mysterious. Here's a cell. A cell does work to create constraints on the release of energy that itself constitutes work. Here's how. A cell does thermodynamic work to make lipid molecules. The lipid molecules then fall into a low energy state called a lipid membrane. That's just a nice low energy state. It's like a marble going to the bottom of a hill. But it had to do work to make the lipids. Small molecules bury themselves into the membrane. You can tell they're small molecules because they might be. And they bury themselves into the membrane, and that constrains the rotational vibrational modes of the small molecules. That in turn changes the potential barriers, going from substrate up over a hump and into product, of the possible reactions that the small molecules can undergo. It really does. Meanwhile, the cell does work to construct an enzyme, thermodynamic work. The enzyme floats over and binds to one of the transition states of one of the reactions that these two small molecules can go, stabilizes the transition state, and lo, that reaction happens with a release of chemical energy. So chemical work got done, and the product molecules come out. But that work propagates because one of the small molecules floats over to a channel protein, uses a high energy source like ATP, binds itself thereby to the channel, induces a shape change in the channel, and that allows an ion to come in which goes over and binds to something else, and work propagates. So a cell literally does the work to construct the constraints on the release of energy, which constrained release constitutes the work that propagates to construct more constraints on the release of energy, which is the work that propagates around to make more constraints happen. Cells really do this thing. I want you to be puzzled because we do not have the concepts for it yet. Now, but what's the task that's accomplished by the work? I'm doing a brilliant job of disordering my transparencies. Okay, we need the Darwinian analysis of function. So Darwin says, what is the function of the heart? The function of the heart is to pump blood. And what Darwin meant by that is that the causal consequence of the heart, for virtue of which it was selected by natural selection, was to pump blood. Fine idea, to pump blood. So the function of the heart is that was selected because it was good for the organism. Now, there are other causal consequences of the heart. For example, your heart makes heart sounds called lub-dub. I know, I'm an MD. We were taught it was lub-dub. It's lub-dub. Okay. Those are not the function of the heart, except for doctors, so they can earn a living. Okay. So there are causal consequences of the heart that are not its function, not that for virtue of which it was selected. That's true. But that means that the causal consequences of a part are a subset of its causal consequences. The functions of a part are a subset of its causal context, and you only know it in the context of the whole. That's true. It's not mystical. The function of the heart's to pump blood. But you only know it in the context of the whole. That means that when chemical energy is released, and chemical work is done, we do not yet know what the task accomplished by the release of energy is. It's only definable in the context of the whole, say the bacterium reproducing. So what's going on is something mysterious but not mysterious. The autonomous agent achieves closure in a set of catalytic tasks, it's collectively autocatalytic, and in a sense of propagating work tasks, by which jobs get done, constraints get constructed on the release of energy that constitutes work, which a subset of the causal consequences of which are the tasks done to construct constraints, including signaling, and the cell literally constructs itself. It doesn't describe itself, it builds itself, or it builds a copy of itself. So there's closure in a space of work tasks. And the mystery that I want you to feel is that it's only definable in the context of the whole. It really is only definable in the context of the whole. So an autonomous agent, or better, an ecosystem of autonomous agents, consists in, try to feel your way into this. I can point to it. It is a propagating organization of work and catalytic tasks that link exergonic and intergonic reactions in a rich web of propagating work, propagating co-construction by which the biosphere literally co-constructs itself, and has for the past 4.5 billion years. Sunlight's falling, there's some hot thermal sources and a little bit of extra sodium and potassium and sulfur and iron around, and here we are. We have literally co-built this thing with the plants and the animals out there over the past 4 billion years. It is co-constructed by us. There is propagating organization, which is the propagating closures that are autonomous agents linked with one another, linking these interactions of exergonic and endergonic reaction. Now, I hope you feel puzzled, because this closure that is a cell that propagates with other cells in a microbial community 2 billion years ago, this is not matter alone, it is not energy alone. You've all been taught entropy, this notion of disorder. It is not entropy alone, it's not 1 minus entropy, namely Shannon information. It is something else, and we do not have the physical concepts for it. I'm pointing at it, you see it, but we don't have the concept. For example, I don't know if this propagating organization is what's called a scalar, namely a number, a vector, a list of properties, a category mathematically. I don't know what it is, but we see them all around us. So the mystery is going to continue. I want to talk about exaptations. Exaptations used to be called pre-adaptations. But Steve Gould called them exaptations, and who's going to argue with Steve Gould? I mean, so they're exaptations. The idea is the following. It's the emergence of novel function of a causal consequence of a part in a new context. So in some context, some causal property of your heart may become functionally significant. And my toy example is, your heart is a resonant chamber, so it might pick up earthquake pre-tremors in Los Angeles, and the survivors would evolve earthquake detectors, because they have to have kids for this to work, okay? So you feel the pre-tremor, you do the right thing, you get all sexually excited, you go out and you have kids. I suppose that's what happens. Why not? Don't laugh. That's where all of the major innovations in evolution came from. That's where wings come from, lungs come from, hearing comes from. And I'm going to tell you a story. I told you I was going to tell you stories. There was a squirrel named Gertrude 4 million years ago, and she had extra flaps of skin underneath her arm and was very embarrassed about the entire thing. And already it had babies, and they had extra flaps of skin. She was up in a tree, and an owl jumped her. And Gertrude saw the owl coming and went, "Ah!" and got away. Now there's flying squirrels. That's roughly what happened. I mean, it might have been a little more complicated than that, but her name was fundamentally Gertrude, okay? And that is what happened. That's where lungs come from. That's where novelty comes from in evolution all the time. Now I'll give you another example. There were some people trying to make a tractor, and they had this huge engine block, and they mounted it on the chassis, and it crushed the chassis. And they mounted it on another chassis, and it crushed the chassis. They kept doing that. And finally somebody said, you know, the engine block is so rigid, we could use the engine block as the chassis. That's an exaptation too, right? Same thing. It's finding a functional use for a causal consequence of a part that you had not noticed before. So exaptations happen in technology. Well, now I'm going to say that exaptations aren't predictable. This is the most surprising step I'm finding myself forced to take. So don't believe it, but look at it. It's an old philosophic saw, but there's no finite description of a physical object. So I'm going to give you a case in point, which happens to be the coffee table in my living room. I've gone back and looked at it, and what I'm about to tell you is true. It's made out of wood, you can't quite tell, but it's got some turned legs. It's actually got a little rod along here. It's got 3 boards. It's kind of old brownish gray wood. It's got a knot there and a knot there. It's got a crack and a crack. By the way, this crack is 1/4 of an inch wide and 18 inches long, and it's about 18 inches from the floor. It's 8 inches from the door onto the patio. It's 256,000 miles from the moon. It's 4.1 light years from Alpha Centauri, and there's a bug just above it right here 3 inches away, which is also about 8 feet from the door, 256,000 miles to the moon and 4.1 light years to Alpha Centauri. And there's a distance from the butterfly to the leg Now you see that I can go on and on and on and on and on with relational properties of bits and pieces of the table to pretty much the rest of the universe. So who cares? Well, I wanted to solve a problem. I wanted to put my power book on the table where my wife and my adult son wouldn't knock it off. And what I did is I took the power cord, I wedged it into the crack, and I plugged it into the floor socket, which I think is pretty good, you know, for a biologist. I did it. That's an exaptation too. Now, after the exaptation, I realized that I could make a fortune going around selling cracks in tables. And I've done that, and I'm extremely wealthy based on that, as you can imagine. So what? The point's the following. That's also an exaptation. Now, the question's the following. Is there a finite description ahead of time of all of the context-dependent causal consequences of things that could turn out to be exaptations? And the answer is I don't think so. So let's be careful here. You can have a finite description of an infinite set of things. Here we go. You remember about sines and cosines in high school. So I can tell you about wavelengths, and I can tell you about phase offsets, and I can tell you about an infinite set of sines and cosines with all the different wavelengths. Fourier, a few centuries ago, proved a wonderful theorem that says if I take a wiggly line in one dimension, I can always approximate it with infinite accuracy by a weighted sum of phase offset sines and cosines of different wavelengths. So that's Fourier power decomposition. So I have finitely specified an infinite set of functions that form a basis set out of which I can describe with arbitrary accuracy any one-dimensional object. My question is there a finite description of a potentially infinite set of functionalities out of which all possible context-dependent functionalities or causal consequences can be built up, having the property that they could be exaptations? And I think the answer to that is no. I'm not sure, but I think it's no. And that's really important. In other words, I don't think we can state ahead of time all of the fortuitous widgets that can come together and do something. Now, if we cannot, then it seems to me that the process of discovery that Gertrude went through with her flying wings, or that we went through, that I did here, is not algorithmic. It's a genuine discovery. It means, I think, that it cannot be deduced from a theory of everything. And we are, in fact, induced to storytelling. We now say something like, after I did that, you see, there's no mystery after the fact. And you could say, did you see what Stu just did? He plugged the thing into the floor socket. And by the way, did you see Gertrude getting away from the owl? We are compelled to a narrative stance in which we say, look at the quixotic unfolding by which things come to exist. There's a story to be told. And I'm going to end with the following. Yeah, I will end with the following. No, I'm going to do two transparencies to give you the flavor of it. Okay, I want to talk about the adjacent possible. Consider all of the types of organic molecules on the Earth as the actual, all the types. Now consider all of the organic molecules not on the Earth, but one reaction step away, as the adjacent possible. The biosphere is continuously expanding into the chemically adjacent possible. We're making new kinds of molecules all the time. We're expanding into the adjacent possible in terms of new kinds of species, or in terms of new kinds of goods and services. So 40,000 years ago, if you wanted to have a rabbit for dinner, you bloody well had to catch a rabbit. I mean, you really did. Occasionally you could trade for one. We can all go out, and if we want to have a rabbit for dinner, or more plausibly a chicken, you just buy one. So something's happened. What's happened is that the number of ways of earning a living in the econosphere has exploded in diversity in 80,000 years. So there's something going on in which the diversity of things is increasing by flowing into this adjacent possible. And now I want to hit you with something very strange. It's that the universe is vastly non-ergodic. It is vastly non-repeating on levels of complexity above, roughly above atoms. So here's how I want, by the way, ergodic means repeating. So you can talk about the Poincare recurrence time of a gas. Let's look at the number of proteins length 200 amino acids, and there's 20 kinds of proteins, 20 kinds of amino acids. How many proteins are there length 200? Well, there's 20 choices for the first position, 20 choices for the second positions, and so this is 20 raised to the 200th. You can tell I didn't do it right. There should be another 0 there. So the number of proteins length 200 is 20 to the 200th. 20 to the 200th is 10 to the 260th. That's the diversity of possible proteins length 200. We could make any single one of them tomorrow in the lab. Just specify it and somebody will build it for you. No problem chemically making any one of them. But there's 10 to the 260th possible. Now let's ask whether the universe has had time to make all of them once. So the universe is about 14 billion years old. A femtosecond is 10 to the minus 15th seconds. There's been 10 to the 33rd femtoseconds since the Big Bang. There's about 10 to the 80th particles in the universe, plus or minus 3 or 4. So the number of pairwise interactions among particles at any one moment of time is 10 to the 80th times 10 to the 80th. And that could have gone on continuously for 10 to the 33rd femtosecond since the Big Bang, and that's 10 to the 193rd, which is a big number. It's 1 with 193 zeros after it. But it's tiny compared to 10 to the 260th, right? That means the universe has not had time to try all possible proteins length 200 once. A fortiori, it hasn't had time to try all possible complex molecules once. It has not had time to try all possible species once. It has not had time to try all possible Chevrolets once, or legal systems once, or languages once. So at a level of complexity above atomic nuclei, the universe cannot repeat itself on timescales that are vastly long compared to the current lifetime of the universe. What that means is the universe is non-ergodic. For complex things to get to exist at all is a triumph. The existence of kinds of molecules, cells, and species driven by exaptations are unpredictable exaptations, and yet what they do is they change the course of the unfolding of the universe. They really do. There were these things called trilobites, and Thomasina was the last one. I know her. And she was about to have babies, and a starfish was about to jump her, and she jumped the wrong way and the starfish nailed her. And now there's no more trilobites. And there's no more unique trilobite proteins, and there's no more unique trilobite small molecules, and the course of unfolding of the universe has changed. Meanwhile, we in our wisdom were fighting with the Russians, and we thought it'd be neat to get to the moon, and we did, and we left off some junk on the moon, because what we're going to do, bring it all back? There's at least one tin can up on the moon of Campbell's soup, plus some other stuff. That's changed the mass of the moon, therefore it's changed the orbital dynamics of the solar system. What we do is consequentful, which leads to the following that I find is very strange, and I'm now going to close. I think the notion of an autonomous agent forces us into a very puzzling stance, in which we have to say that such entities exapt and discover new ways of making livings, new ways of making livings that could not be deduced ahead of time, but come into existence and change the world. There really are flying squirrels. There really are lungs. These really rare things exist and change the unfolding of the universe. About them, ahead of time and afterwards, there's a way in which all we can say is, wow, did you see what Gertrude pulled off? We are called to a narrative stance. Watch what just happened. That's wonderful. What is it then to do science? Well, we've been doing it as biologists for a long time. So there's a lot of things to do with science, and if we had a little time longer, I would tell you some more about it. But among the things we can do with science is we have to ask what it means to do science in a fundamentally historical realm. And biologists have been doing it for a long time. We can ask, are there general laws? I think there are general laws about the co-construction of biospheres, and it would be another 20 minutes to tell you those potential laws. We're invited to a new marriage of C.P. Snow's two cultures. How do you do science when you tell stories at the same time? But we do have to do both. I'm sorry, because I feel like I want to tell you a bit more about some of these laws, but fundamentally they say that the biosphere may be self-organized critical. When you pile sand on a table in pair box model, you keep piling it up, and then you get small and large avalanches of sand piles, sand slides. And I think there's grounds to think that you get small and large avalanches of speciation and extinction events in the biosphere and in the econosphere, where they're called Schumpeterian gales of creative destruction. So I think there's all kinds of general laws to be found, but at the same time, the individuals who are doing it have their stories to tell in that unfolding. So I'm going to end with that. I think all of this is very puzzling. And so you see, I started with Manna because she grounds me in something that people knew 80,000 years ago and we've forgotten. Namely, we are rooted. And we're rooted in a biosphere that we're co-constructing. And I have the feeling that there's a spiritual message in all of this too. It's strange that Vice President Gore ended on a spiritual note. He asked, what would be the meaning to us if we discover life elsewhere in the universe? And we will, or we'll make it here. So we will be at home in the universe and natural expressions of it, but we are co-constructors of the universe that's around us and the biosphere that's around us. The universe on this view is creative ever because of exaptations that cannot be pre-stated, but afterwards, wow. And you know, this is a secular age, and it's a plastic age. How do we feel if we are expected in the universe, or something like us is expected, and if we are responsible, but if we never know the long-term consequences of what we do, and we never have and never will? So when Manna and her predecessors were young, 3 million years ago, somebody picked up a rock thinking about the analog of a coconut, and then thought about Fred's head. And it's been like that ever since. Thank you. I'm sorry I went on over long, but not very. And thank you all for being patient and staying. And I will be glad to take questions or comments or battles. Yes.
The consequences of much exaptation must be on the level of significance of the conservation of your PC book.
You mean not too important?
Not too important. In fact, it seems to me there must be an ocean of inconsequential exaptation, out of which only a few incidents arise where flying is important to squirrels.
Yes, that's right. What's interesting, of course, is that we know this. We know this because it's not part of science speak. It's part of our ordinary knowledge that, most things don't matter, but some things matter a lot. So we might be able to get laws that are statistical distributions of the probabilities of events having small and large cascading consequences. For example, my friend Brian Arthur at the Santa Fe Institute points out the following. He says, when the car comes in, it drives out the horse. And with the horse goes the buggy whip, the saddlery, the pony express. But the car paves the way for paved roads, gas stations, traffic lights, traffic courts, traffic cops, bribing traffic cops, fast food restaurants in suburbia. So that's a pretty big and consequential change. In fact, it's a Schumpeterian gale of creative destruction, with the notion that there are small and large Schumpeterian gales of creative destruction in the economy. But the hula hoop came and went, and who cared? So, well, I didn't really mean that. But I mean, not as many people cared in as many different sectors of the economy about the hula hoop as did for the cars coming. So somehow we need a theory. You see, here's the case where there are stories and there are stories. The detailed explanations of any one of them is a story. On the other hand, some of them have lots and lots of consequences. It's a real good thing we beat Hitler in World War II. But it's not quite so important that I got stopped at a traffic light. Right, so we need a theory of the we can do science by asking about theories of the distributions of propagating consequences. On the other hand, there really is history. It's very strange.
Thanks.
You're welcome.
And then over to the right.
Yes.
Yes. I was wondering, would you speak to the possible dangers of trying out all these new proteins?
Serious. It's something I care about a lot because I have one of the earliest patents in it. And the question is, here we are where we can make the molecular diversity of the biosphere and beyond. I mean, people are now making about 10 to the 18th novel molecules. Well, ricin is out there and it's a pretty bad molecule. So can we make an analog of ricin? Well, almost certainly we can. The hope is something like the following. There's grounds to think that the number of effectively different molecular shapes is about 100 million to a trillion. Because molecules only interact with one another over a certain size scale, and they have to be, there's a sort of an effective sloppiness in molecules recognizing molecules. And your immune system has about 10 to the 8th different antibody molecules, which kind of covers a shape space. It's sort of universal in its capacity to recognize, which means that there aren't gazillions of utterly novel things to make, probably. But it doesn't mean we can't make bad things. We are confronted here, as we are in all sorts of areas, like with atomic weapons and other things, where we have something where we do not know how to assess the risk. It might be catastrophic, but it seems remote. What will we do? And what we always do is go ahead and try to be sensible. And I think that you're immediately picking up what I think is the profound moral problem. We do not know the long-term consequences of what we do, whether it's with picking up a stone 3 million years ago and thwacking somebody on the head, or this. We never know in the unfolding universe and in the unfolding unto an adjacent possible. We cannot calculate the consequences. So we have to struggle with what it means to be responsible anyway. So I don't think there's a simple answer to what you're saying. We will do what we always do. We will do something about being cautious and taking adequate care and trying to think about it. And we better, because we could do bad things. And then we'll go on. Yeah?
That was almost exactly the same question I had. And I was going to formulate another theory that I think life is very common in the universe. It started on the Earth very quickly.
Right.
But I think intelligent life might not last very long, and it has to do with the inevitability of people playing around with things like atomic bombs and new viruses, and that complex systems will eventually have some sort of an explosion.
Yeah, we'll nail ourselves. Yeah, I don't know what to think about that. I think we all think that cockroaches will survive, right? Deservedly so. I just don't know what to really make of that. It depends upon something about, I don't know how you assess something like that. Carl Sagan's view and others' views were that we'd really bobble ourselves. It's going to be pretty hard to wipe out the entire human species. We're the most abundant large things around, even more abundant than cows. The bacteria will win. I don't know. I don't know how to assess that. I share the worry. Yeah.
Well, somewhat along the same lines, I haven't been able to formulate a careful question, so I'm going to give myself 33 seconds and give you kind of a ball of stuff to just wrap around. But this thing of increasing complexity, it seems like it's not the creation of an anything goes atmosphere when you do that there's a corresponding sort of implosion in your degrees of freedom, where I have a sense that as it gets more complex, I'm less willing to let you just do anything. Because of my sense of the equilibrium, the whole, and what your function is, for example. Mm-hmm. And then I have the question as to why I use the word exaptation, which seems highly technical compared to synergy, which I know is old, overused, hackneyed, but still very pop. We could all pick that up, whereas exaptation doesn't have that. And then I would finally, I'd say, language is a very important constraint system. So like if you don't stop using exaptation, you won't get that $5 million from NASA. I'll see to it. I'm just saying it like that. Okay.
Could you please count up the double negatives in that? I want to come out on the right side of that one.
I understand. I'm all for you. I think you're a great guy, and Gore should win the election. Thank you.
Thanks. Well, let me try exaptation first. It's not just synergy. What Darwin was pointing to in the notion of pre-adaptation, and Steve Gould has called exaptation, and who are we to argue with Steve, after all, is the idea that a causal consequence of a part that was not functionally significant can become functionally significant in an environment. So my toy example is your heart managing to pick up earthquakes, and now there are earthquake detectors. And the point of that, which is not merely the point of synergy, is that in the new context, it is genuinely the case that it's useful. It's a nifty thing to use an engine block as a chassis if whenever you put the engine block on a chassis, it crushes it, but you've got this big massive engine block that's rigid. So I need a notion like exaptation because I'm so struck by the fact that you cannot deduce it ahead of time. You can't deduce it ahead of time. It's a genuine discovery. So the coming into existence via exaptations of Gertrude the flying squirrel and tractors is the universe being creative in a way that wasn't statable ahead of time, and it's because somebody can make a buck at it in one way or another. Gertrude gets to have kids, and whoever came up with the tractor gets to proceed that way. So I think we do need a different notion than just synergy. Could you remind me of the first point that you made?
Well, actually, on that last point, it seems that the literature I read, the unpredictability is precisely what's emphasized with synergetics, and that if you want to use exaptation, it seems like the work's already been done, and so you're kind of being redundant with existing work, and that's why I have my problem. But that's all I have to say.
Could be. You also had a first point you were raising, though. Do you remember? Can you remind? Okay, we'll go on. I wish you remembered it. I had a brilliant response.
Okay, you may.
Oh, good. Where?
Go ahead.
Oh, yo.
Yeah, what are your thoughts on how the second law of thermodynamics could be possibly expanded to account for complex organization?
Well, with lots of hesitation, I do have some tries at it. So investigations starts by asking, could there conceivably be something you'd want to call a fourth law of thermodynamics? And of course, I don't know the answer to that. But I'll try to tell you its theme. The universe is non-ergodic, if I'm right. My claim is the universe is non-ergodic. It will not repeat itself on vastly long time scales. So we could ask if we can define an adjacent possible, which I think you can do, and I think you can do it rigorously in both classical systems and in quantum systems, then you can ask whether or not the universe arranges itself, or a biosphere arranges itself, something that, in a way that governs the way it self-constructs and flows into the adjacent possible. The first thing to be said, by the way, is that there could not be a general law for all possible non-equilibrium systems. And that's easy to prove, and it goes as follows. There's a first-order infinity of possible computer programs. Any one of them can be realized on the computer by programming the computer and plugging it into the wall. So you can make a real non-equilibrium system do something that you can program. Most computer programs have the property that they behave in ways that you cannot foretell. You can only run it and see what happens. Just try it. Okay. Now, if what you mean by a law is a compact description that allows you to say ahead of time what the system will do, then most computer programs are not lawful in that sense. There's no compact description ahead of time that foretells what it does, and so there could not be a general law for all possible computer programs, hence all possible non-equilibrium systems. But in that case, notice it's we who came up with the funny program that we jammed into the computer. which leaves open the possibility that for co-constructing things that come into existence with our hands off, namely a biosphere, anywhere in the cosmos, there may be very general laws about what is co-constructible. And I have the hunch that, in fact, it does relate to the last gentleman's last question, or first question. Think of, in the econosphere, think of future shock. That's supposed to be the fact that goods and services will innovate and come into existence so fast that we can't keep up with it and will be overwhelmed. Well, that's silly because, of course we'll keep up with it. If we couldn't keep up with it, we wouldn't make a buck doing it. So what happens is that we will introduce new goods and services at that rate at which our organizational structure can master the problem of introducing the new goods and services for the life cycles of those goods and services, whereby we can make a living. In the biosphere, I think it's the same thing. There's a maximum rate at which functional novelty can make its way into organisms. If novelty intruded itself faster, natural selection would not be able to keep up with the encroaching novelty, and it would destroy all the organization. So there has to be a balance between the accumulation of novelty and the correcting of it by something like natural selection, so that critters can make a living, so that bacteria have new ways of doing new metabolic things out there in the sand pile or the toxic waste dump. So I think that what may be going on is that the entire biosphere is sort of self-organized critical and gates its entry into the adjacent possible in a way, I'm hoping, that on average maximizes the rate of encroachment and expansion of this adjacent possible. Now, if ever one could show something like that, that might qualify as something like an extension to the laws of thermodynamics, to non-equilibrium systems. So I've got dreams in those directions, but not theorems. Thanks for asking.
So over here now.
All right. You mentioned that the universe is non-ergodic only above the atomic level. Why is that?
Well, it's actually non-ergodic at the atomic level, too. It's at least the case, if I understand what my physicist friends are saying, that you can calculate all stable nuclei or all modestly stable nuclei. But in fact, the most stable nucleus is iron, and there's an awful lot more hydrogen and helium in the universe than there is iron. So we are not at equilibrium with respect to the matter distribution in the universe because we're not chock-a-block full of iron. For example, most stars are hydrogen and helium. But at least, presumably, we've explored all or almost all of the reasonably stable nuclei at least once, because there's only a few hundred of them. But if you think about proteins and large molecules, the number of possible large molecules is so vast, like 10 to the 500 or 10 to the 800, or you concoct what you want, that there simply has not been time to try all of these combinatorial objects one at a time. Okay? Yeah?
That leads exactly to my question. Why one at a time? Why not multiple? I mean, life forms.
Sure.
We have multiple systems.
Yeah, and the point of the calculation that I showed you was that if you take 10 to the 80th particles bonking into one another every femtosecond, which would be as massively parallel as possible, so all 10 to the 80th particles hit all 10 to the 80th particles in pairwise collisions every 10 to the minus 15th second since the Big Bang, which is not one at a time. Okay, there could have been a total of 10 to the 193rd pairwise collisions, or something on that order, which is a very crude calculation, which is trivially small compared to the number of possible molecules to be constructed. So it's not that you're doing it one at a time, it's that the universe, even acting in parallel, cannot have made them all. So, the universe just has to be non-repeating. It's a very strange thought. It has to be.
Okay. I just didn't understand that point. Thank you.
Okay. Of course, I should tell you that I've given this talk in front of some pretty good physicists, and I haven't gotten blasted yet. But it doesn't mean that I won't. Yes, up above.
Do you possibly see the emergence of something that is not as specialized as mathematics, describing some of these insights, but not as vague as English, with all the areas where it can just persuade and mislead? And I mean, this is something that I've been seeing more and more in your field, and what has drawn me to complexity and chaos is that these are insights that we all have on a visceral level and say, yeah, that's it, but find hard to express it. Sometimes we can express these things via mathematics, but it takes a long time for their applications to be present in society where your common person understands what's going on or is participating.
I very much hear your question, you know, and I've written 2 books of stuff that is mathematics and well formulated, or at least reasonably well formulated, and is more or less standard science. If questionable or whatever, at least one knows what it is within the terms of ordinary science. The stuff about autonomous agents has me profoundly puzzled. And the reason it has me so puzzled is that I can see it. I can see the context-dependent closure. So can you. Once I've told it to you, you say, oh yeah, there's one. The problem is, if one wants to, there's two places one would like to mathematize it. One would like to have a mathematical description of what such a closure is. Well, there's a way of doing that in what's known category theory. But the problem is that even category theory, as far as I understand it, has the problem that you have to be able to state ahead of time all of the possible mappings. I think you do. But it doesn't seem like we can state ahead of time all of the possible mappings, because who knew about Gertrude and the flying squirrel? So if we can't ahead of time state all the possible mappings, it's a little bit like Gödel's theorem, or feels a little like Gödel's theorem. Well, you know, Gödel's theorem says to us, golly gang, you can have a set of axioms and there are statements in that formal language having the property that if they are true, they're not provable. Namely, you can't derive them from the axiom, so they're formally undecidable. But then if you add a new axiom, formally undecidable statements become decidable. Namely, you can derive them as theorems, algorithmically, but there are still some new undecidable statements. I kind of have the feeling that this thing that I'm struggling with is a little bit, see, where do the new axioms come from when the mathematicians do it? Well, they're smart because they're mathematicians, you know, because mathematicians are smart. And they pluck them out of the air and say, here's a nifty new axiom, and now new things happen. In the universe, it's not that there are axioms, there are exaptations that unleash new possibilities, hearing, for example, and then new things can happen. So I have the sense that these exaptations are a little bit like adding new axioms, but the propagating organization that is a biosphere builds them and discovers them and unfolds into them, even though they couldn't have gotten said ahead of time. But when they come up, the universe can behave in a new way. For example, we have forward planning horizons. We can make models of our world, and once we can make models of our world, the way we behave is different than the way, say, viruses behave, and different things happen. For example, stock markets with forward planning horizons and speculative bubbles come into the universe, but could not have been there for agents that do not have forward planning horizons. So it's just incredibly strange. Phil Anderson, this is the Nobel laureate who told me that work is a strange notion, wrote a paper 20 years ago saying more is different. And I think this is part of the unpacking of what Phil is intuiting. And it says, if it's right, that there's something that really is emergence, that's not mystical, it's real, it's in our storytelling. It means that we've got to do science in a new way. You see, Newton taught us how to do science by saying what the variables are, what the laws are, what the initial conditions and the boundary conditions, and now you crank it out. But Gertrude the flying squirrel is not a question of what the variables are, it's the coming into existence of a new kind of thing called Gertrude the flying squirrel. And that's not in the story that the physicists have to tell us. I do not understand this. Yeah.
Just have a couple of short questions. I work in the state legislature in Salem, and it's rather discouraging to think that. Well, let me just frame this. And the first question would be: do you think that it is a general law that to maximize encroachment into the adjacent possible?
I suspect that the answer to that's yeah. And I think we can find out if it's right. And I'm struggling pretty hard to try to formulate what it would be to describe that and do it properly. But it has to be in some sustained sense. You see, suppose that you took all of the organisms of the world. I'm going to tell you my great Noah's vessel experiment. You take all of the organisms, all of the species in the world, there's 100 million, and you take them 2 by 2, and you put them in a vessel, but it's not Noah's vessel, it's a big blender. Okay? It's a biochemistry experiment. So you normalize, you put whole fleas in, but a tiny scoop of hippopotamus stuff in. And then you get this big vat, 100 million, it's got to be pretty big. And then you do the really following subtle experiment. You gush it all up. I was in a department of biochemistry. That's what you do. You gush things up. There's more technical names for it, but basically you gush it up. So you're mixing the sap of life with the sap of life everywhere. I think if you did that, and I didn't show you, but I showed you the button in the thread story. I think if you did that, there'd be so many molecules and so many potential reactions and so many potential catalysts that you would get an enormous explosion of molecular diversity. A supercritical explosion, but it would also destroy everything in the system. It would rip it apart because you'd make functional components that if ricin is a worry, it would just tear apart all of the organization. It would rip into everything, okay? So what has to be happening is the biosphere and the econosphere have to gate its way into the adjacent possible such that it constructs itself and propagates organization at that rate there that we can make a living at it. So if there is such a law, we're going to have to formulate it pretty carefully. And also, even if it's a law, it's not naturally and obviously right. For example, there have been cultures that have put off any sort of cultural and technological innovation for years. The Japanese culture did in the 1400s, and Islamic culture did for hundreds of years, and so on. So it's...
Well, that was my follow-up question, was that in your example, you sort of perceived in your mind that you could put the cord in the crack before you actually did it. You could see what the use was before you did it. And maybe, and like the example with Japan banned firearms and only allowed swords, and that could be a good argument for, not being as many murders in that society at that time. Maybe there were more murders by swords, I don't know. And I guess what I'm getting at is, do you think that it's possible for mankind, from a governmental perspective, to limit that expansion, that unbridled expansion into the adjacent possible, and use that so that it's not quite as scary to, allow genetic, all these things to happen?
Well, let me take it technologically. By the way, the fun part of the story about Japan and the swords and the firearms is it was the samurai who disallowed firearms because they didn't want second-rate dweebs from the port cities coming up, with guns and blasting away. And so it was sort of a technique to maintain power as a samurai warrior class. And so they didn't know about Saturday night specials, but they foresaw them. So you see, I think meanwhile what's going on, and this is what I was pleased to and honored to be asked by Vice President Gore to write about, is the following. What's driving economic growth is, in fact, the economic web. The economists are making single-sector models of the economy in which there's one thing, it's called stuff. That's technical, and it's purple. It's the gross national product. And the idea is that economic growth is happening because you increase labor and capital and the product of them. You got more machines and more people, and then you make more stuff. Then it turned out that that didn't fit economic growth, and Solow put an A in front of it. So it's A times labor and capital, where A now is human learning. And that's supposed to be what's driving economic growth. We're getting smarter and smarter and smarter. It turns out that any single sector saturates, and if you look at power turbine output, for example, it goes up and saturates. And if I'd given another talk, I'd have talked about fitness landscapes and the fact that you always saturate, because the better you are, the fewer ways there are of getting better. And that's a whole nother talk. So what's really driving growth, I think, is the economic web. The fact that there are goods and services that are niches for other goods and services, complements and substitutes. Screw and screwdriver are complements, screw and nail are substitutes. New goods enter the economy in the niches created by old goods and services, and I think niche creation's autocatalytic. So the more goods and services you've got, the easier it is to make new goods and services that fit into it. That's why I think that the biosphere is expanding into the chemically adjacent possible at an ever faster but gated rate. The more kinds of reactions you've catalyzed, the more ways there are to do new things. That's the same thing is true for morphologies. There's a secular trend to increase the diversity of species, according to the best experts. And it really is true that the number of ways of making a living have gone from a few dozen 80,000 years ago when mana was first carved, to millions now. So this advance into the adjacent possible is forever constructing new niches and new ways of doing nifty things. And I think it's wonderful. And I don't think we should be terrified of it. We just have to be wise about it, whatever that means. You got a lot of jobs to do in the legislature. Don't blow it. Maybe a couple more, yes.
Well, I was curious if you've given any consideration to, or have any theories on what or how the effect of, if evolution finally does get around to testing all of the different proteins and forms of life that can be made, what's going to happen then when everything's been tested if it can't repeat?
See, I don't think that everything can be tested seriously. Just again, think about, if you just think about the number of possible proteins and DNA molecules and RNA molecules, let alone morphologies, I don't even think you can, I think you can at least formulate the set of possible proteins length 200, because it's 20 to the 200th, so you stick yourself with 20 amino acids. I don't even know that we can count what we would mean by the number of possible self-reproducing autonomous agents, because I don't know how to pre-state what I mean by all the possible functional closures that can work. So I don't think the universe is ever going to manage it. So I don't know how to answer your question. Sorry. Yeah.
My question, can you hear?
Last question. Okay.
My question stems from, I guess, my awe at the human organism, or you know, how developed we are as far as having lungs and hearts and such. And you mentioned in several of your models, from the E. coli bacteria swimming in a sugar material to also your model of autonomous molecular autonomous agents, and then brought up the button theory of, or just having many molecular agents interacting with each other. Do you see that there'll have to be just a sea of different sugar-like mediums to support such a interrelated activity, or will it be something else, that there are so many things making different molecules that are part of other cycles that it'll either become easier? I guess, would it become easier or harder for maintaining such a sea, I guess, of activity?
Wow, what a neat question. And one might ask that question for a microbial ecosystem or some analogous system. Roughly what we want to ask is something, here's what I hear you asking. Consider a general biology for the emergence of life anywhere in the cosmos, and homologous to that, something for economic systems, which are just generalized forms of ecosystems. Now think about the energy resource space that supports it, because that's what your sugar molecules are, right? What's the relationship between the diversity of the energy sources, the diversity of the species, and is it the case that as the diversity of species increases, or critters increases, it gets easier rather than harder, okay, to somehow make use of whatever energy sources are around? I don't know the answer to that. I think that's a wonderful and profound question. I have an intuition. I think it gets easier because there's sort of more and more places to plug in a particular source of free energy into this big web. And there's evidence that suggests that your intuition, which says it gets easier, which is I think what you're thinking, and mine, is correct. And that evidence is that if you think of the residence time of energy in an ecosystem, you can tell that there's energy in an ecosystem because if you take a piece of wood and burn it, it gives off heat. So there really is energy stored in an ecosystem because you can cook an egg. There's grounds to think that ecosystems, as they mature, store energy over longer and longer and longer time scales. So they're getting good at pumping themselves away from equilibrium. And my own feeling about this is something like this. I don't know how to prove this yet, but the more the system pumps itself away from equilibrium, for the very reasons we saw, doing work cycles and linking exergonic and endergonic reactions, the more alternative macroscopic arrangements there are of doodads, like my pipe watering the cornfield. Now, I'm not sure that this is right, so put a big caveat around it, but if that is true, then the more possible alternative macroscopic arrangements of widgets there are, the easier it is to find widgets that propagate the constrained release of energy that achieves closures that are self-sustaining wholes that propagate and can sustain themselves, so that somehow it becomes easier, not harder. And that might be tied to the sense that one has that the global economy is on a point of just an explosive takeoff in creativity. It's easier and easier and easier to create value, not harder and harder and harder to create value. There are so many frontiers on which we can do nifty things. So probably in closing, the thing to say, what a wonderful mystery all of this is, how far we are from Darwin and natural selection and random variation, a piece of the story. And if this notion of autonomous agents, exaptations, and the non-capacity to state ahead of time what it is right, and I don't know that it is, how true Newton was for us, but how far we are from it. He never could have gotten us flying squirrels. So that's awesome. We live in an unfolding creative universe. So the spiritual implication may be that we discover that that's right, and our view of ourselves in the universe becomes one of membership, co-constructing responsibility. And that will be very, very interesting. Be a neat millennial quest. Thank you all.